couple to have children, and then the rest are donated for research if there are excess. So it's a very different system in the UK. But I think Maria's criticism was more to do with the lack of the double tier of regulation and control, I think, rather than...
Yeah, it's a very local regulation, and again, I don't know the details really, but you know, in a way there's a conflict because the institution's research is being supported by outside donors, and so if they shut down their own research, then they're losing donations, and so it seems to me you need a more independent oversight than is seen in the U.S.
It's a similar. I mean, I understand it's a similar issue in China, for example, because it's all local ethics approval, and there's no other checks or balances.
So for me, I don't think I face this kind of issue because I collaborated with my IVF collaborator. He deals with every concern about that, and also he get the IVF donor once he have like extra sperm. Then they will discard the zygote, so they can use that for analysis. Totally different. So.
The second question, second part of your question.
Yeah, I mean, again, it's interesting that the exceptionalism under which the NIH operates with respect to embryo and gamete research has created the kind of void that you've discussed. I mean, absenting themselves and then creating an entrepreneurship among researchers is really responsible for the kind of void that you've described. And there are many who would like to see some movement in that area, and so I just wanted to pick at what you were critiquing as to the outreach that researchers, including the Oregon Lab, have engaged in essentially a crowdfunding sort of manner, is really a response to the way in which much larger decisions have been made at the national level.
Exactly.
All right, thank you so much.
Hi, my name is Tiago Fayal. I'm an editor for Nature Genetics. So I cannot comment, obviously, on what happened at Nature because we're editorially independent. But if you want to talk about that afterwards, I'd be happy to talk a little bit about that. That's just a brief comment. And then the second is it's a scientific question, which is on what Kathy mentioned. So you briefly mentioned that human embryos, by and large, have a relatively high genomic instability. And I guess my question is, do we know if that's because our IVF culture manipulation conditions are deficient, or do we know that, do we know anything about the level of genomic instability in vivo, in humans, or even in other mammals besides the mouse? I mean, that's part of the question. And is that a problem in the sense that, I mean, there are data from, Magda Jonice-Goetz and other labs suggesting that cells with genomic abnormalities might eventually be selected against, during embryogenesis. And of all these, these two issues, what would be the impact for mosaicism in selection for, genetically modified human embryos?
Yeah, these are all really great questions. In an IVF clinical context, most embryos that will arrest around the 8-cell stage, which is coincident with embryo genome activation and that first checkpoint I mentioned. So the very first time an embryo senses that something has gone wrong is at around EGA, which is interesting because why does it even continue to develop, even after that catastrophic event? And so the high incidence of aneuploidy that's detected, that's the first bottleneck. And the embryos that develop beyond the 8-cell stage, from work from Dagan Wells, people who are the leaders in looking at karyotyping of human embryos, all suggest that if they develop beyond the 8-cell stage, their incidence of aneuploidy is actually quite low. And if they are aneuploid, it's invariably mosaic. And one of the hypotheses is any cell that's quite karyotypically abnormal might be tolerated in the placental progenitor cells, but not in the embryo compartment. So if it did have it in the embryo, it would have kind of arrested before that. And then in terms of other organisms,
Well, maybe I can mention one thing here. It's known that human females have very poor success or have very common abnormalities after meiosis, and can manage to get through the meiotic divisions, and so there's something very inherently error-prone in the human female, much more so than in the mouse.
And I think it's exacerbated by poor culture conditions because, again, there's this 8-cell stage kind of, the vast majority of embryos will rest around the 8-cell stage, which also is coincident with EGA, and that's in the mouse in vitro fertilization developments from Anne McLaren and John Biggers, that would be associated with the 2-cell block in the mouse. So the equivalent of the embryo genome activation in humans that occurs at the 8-cell stage happens at the 2-cell stage. That was associated with very poor culture conditions, and as soon as they developed those refined techniques, then they had really high rates of blastocyst development. So I think that's the poor culture conditions undoubtedly, I think, are exacerbating these high rates. And it's also quite high in non-human primates. But again, one doesn't know whether it's an intrinsic. I think it's probably a combination of intrinsic and also poor conditions because we just don't know enough. And a lot of our understandings are from rodents, which are, I think, perhaps not quite the same. Yeah.
Thank you.
I have a relatively simple risk-benefit technical question, and it's basically, is the risk of going through IVF worth the possible benefit of knocking out a gene like CCR5 or correcting beta thalassemia? And this question doesn't really come from me, it comes from a neonatologist that I had a conversation with at a dinner party, and she'd never heard of CRISPR before, but she said that a lot of the babies she sees in her clinic are products of IVF, and I think actually I was encouraging Bill, I was goading Bill to ask this question with more technical sophistication about some of the problems with IVF, but basically I'm just trying to understand, it seems like going through IVF does present a degree of risk, perhaps a significant degree of risk to the unborn child.
Well, I think, actually, Paula might have much more to say about this.
But, ask it better than me.
I actually just came across a Washington Post article that talked about the lack of regulation of IVF. It's almost like another version of what I was talking about earlier. So there's not a lot of oversight to really, track all of the kids and such, and that's kind of a shame because you can't collect data. But obviously, there are lots of human children that are perfectly normal after IVF, but whether they're at significant risk or not, I don't know.
I mean, I don't know whether you want to expand on the question, but I can address a little bit of it. So there have been some large-scale studies where, out of Australia, for example, and in the UK and other collectively through European Society for ESHRE, yeah, and ASRM, and the evidence generally is that if there is a problem for having children with IVF, it's quite small, it's minor. There are some suggestions that certain procedures, like ICSI, for example, PGD, might have a subtle effect, may have slightly more errors. But it's always hard to do these studies because, of course, you're having it's a selective group of individuals who are being analysed who have problems of fertility and who may be older, so there are other issues there, which makes it very hard to actually know really what's happening and whether any. I mean, maybe Helen can comment, but you're going to comment in your go. Okay, why don't we leave it to Helen to give that answer properly because she's associated with this type of thing. So it's just hard to get the data.
Maybe I should clarify what we were talking about here. So last night I was mentioning that in my conversations with JK, that's Dr. Hay, I don't know how you say his last name, the scientist we heard from yesterday in any case. JK and I had long conversations together when he came to Stanford. And we talked, and here it was theoretical. He talked about how he would do various procedures. And included in that was the idea that it might be good to give people CCR5 so that they were protected against. And I said, well, you would do this to a normal, otherwise not at-risk person? And he said yes. And I said, well, are you aware of the fact that there's a low rate, but there's a very real rate of downside to IVF itself? And would that would you take that risk in order to confer a theoretical benefit? That's where this conversation is mentioned here. And I'm not an authority on this, but I followed the literature on it, and there's a 40 to 60% higher rate of congenital defects. For those of you not scientists, congenital does not mean genetic, it means at birth. And most of them are fairly minor, but some of them are very significant. There's some very severe epigenetic diseases that come from this, as you know. And then, as you say, with specific disorders, with specific procedures like ICSI, there's higher rates of things like hypospadias. And so my only point here was, in talking with JK, that you don't do a medical procedure on a healthy person if you can avoid it. And I raised that not knowing he had already done it, but knowing that, but theoretically that there were contraindications for even entering into the realm of unwarranted.
I think that's a very valid point.
Yeah, so that was my point. And just so, because it came up, let me just add this. The issue of aneuploidy, it's a very interesting one because the thinking has gravitated a long way on that thought about whether to do PGD, and the famous story now, it's public, about Magdalena Zernicka-Gertz carrying an aneuploid baby and not being willing to abort the baby, but then starting to study the migration of the cells. And it's a very positive story. The baby was born healthy. But the interesting thing is, and this is another cautionary concern for our interventions in human life, there's at least theoretical evidence that the aneuploid cells migrate to, maybe not just sustain, but maybe actually actively migrate to the placental tissues, the extraembryonic tissues. Is that true? And if so, there's also at least some discussion of the possibility that aneuploidy assists in placental efficiency, and I mean, I'm a physician, so we go by above all do no harm, and I think it wasn't said once here in this conference that we need to always remember that every procedure of intervention comes with potential unknown unknowns, is the famous phrasing.
Can I just comment about Maria's the part of your talk which was about publishing and the problems there? I'm going to declare a conflict of interest because I'm on the board of Public Library of Science, the PLOS Journal, and of course we promote not just open access now, but also post-publication peer review, so submission of all articles onto bioRxiv, for example. And that gives an opportunity, of course, for others in the community to post comments about papers or manuscripts that have been posted and to criticize them, and you can get a rapid response. So I encourage everyone to do that. But as I say, I'm conflicted.
I have to say, bioRxiv for me was really instrumental for getting out our message very quickly, and one doesn't want to use it as a mechanism to criticize every published paper, but it certainly was a really useful venue for us given the year-long wait at Nature.
I think we should go to panel 2. Thank you. I've lost my... You're next. So I lost my program. So our next speaker is... Oh, you need to load it. You need help.
You're here.
Oh, I see. Okay.
Just
well,
might
so
take a while because it's a large panel. So.
All right. So while Helen's slides are being loaded, just introduce her briefly. So she's at University College London. She's works closely with, Women's Reproductive Unit. What's it called?
Institute for Women's Health.
Institute for Women's Health. Sorry. So she's very knowledgeable on matters to do with IVF and assisted conception. And so she wants to give us a few ideas on forging a bench clinical pipeline in human germline editing.
Thank you. Thank you for the opportunity to speak. I'd like to first point out that if you make a base edit to the title of my talk and do a G to C conversion, you get forging changes to forcing a bench to clinical pipeline. And we've already seen that when you force a bench to clinical pipeline, all sorts of off-target events happen, like this week. Now, mention has been made of the Asilomere Conference in 1975, when recombinant DNA technology first came out. This idea that if you put DNA from one organism in another, that there may be some unintended consequences of a technology like this. This asexual reproduction, as the microbiologists would have called it. But today we can celebrate so many of the advancements of recombinant DNA technology, and we take many of our treatments for granted, and even including our vaccines for granted, not just recombinant, but synthetic DNA technology. Another technology that had its birthday 40 years ago as well was IVF. And so we've mentioned about IVF, but this month marks the birthday of Louise Joy Brown, who was born 40 years ago as the first IVF baby. You might say that this conference is a meeting of two 40-year-old virgins. So while these headlines kind of show a very positive aspect to IVF and the celebration of this lovely Louise or super babe, there was certainly negative undertones and major catastrophic backlash in the press and in the media and among other communities. Many of those communities have still not accepted assisted reproduction. And we see an image here of Patrick Steptoe and Bob Edwards in the lab, and a misconception that Louise Brown was a test tube baby when she was actually incubated in this jar. And it really gives us an idea of where the technology has come. We have incredible instruments now, not only to harvest oocytes, but to culture embryos for extended periods, to incubate them at the appropriate conditions. But I also want to make note of the fact that when Louise Brown was born, their clinic was closed for 2 years. It took them 2 years before they were able to do any more experiments. And the only reason they were able to do this was through privately funded donations from people and from themselves before they were able to go to Bourne Hall. And this was a post from the New York Times from a very renowned scientist, James Watson, who predicted before Louise Brown was born that all hell would break loose politically and morally all over the world. Now, little did he know he was kind of correct, but that has more to do with Trump and Brexit than it does to have to do with any IVF babies. And I want to draw your attention to the second IVF baby, who was born just 67 days after Louise Brown in India. Oh, I don't have the picture. But the doctor who created the birth of the second IVF baby, I wish I could pronounce his name, it's very long in Indian. He actually received such criticism and such rejection for what he had done that he committed suicide in 1981. So I think that highlights the different cultural acceptances of certain technologies and how we can move, and how sometimes something can be catastrophic to someone's career, and sometimes in another cultural setting, it can be accepted and very much celebrated. So in talking about developing a bench to clinical pipeline, really we're talking about the idea that there may be an entirely new field that would be accepting a CRISPR clinic. And actually what I want to try and talk about today is a little bit of context about how easily a transition could be made to the clinic. And the clinic might look something like this. So this is a drawing of a very modern clinical embryology lab. And you'll see that there are different parts which are for procedures and different parts which are for just the embryology lab. So there will be different flow mechanisms for clean air, for just the embryology staff, so they would have ultra-clean facilities. But most importantly, then, on top of all of this state-of-the-art facility, is that it would adhere to the guidelines set out in directives such as the European Tissue and Cells Directive and the HFEA Code of Practice. So on top of having an already quite comprehensive laboratory in which these experiments could occur, they are also underpinned by very critical laws, which mean that their standards have to be at a certain practice, that the storage or procurement of gametes and embryos are also regulated and protected. And the current clinical pathway of assisted reproduction, I just want to highlight that actually when we think of the field of assisted reproduction and we think of germline editing, really there's so much more of a negative thought around germline genome editing and what we could be potentially doing to an embryo. And I want to look at all of the different processes and the pathway of assisted reproduction. There is nothing different about what we would do to an embryo if we were doing germline editing in terms of the structure of the embryo as we would in the normal IVF laboratory. We would still prepare sperm. There would still be oocyte retrieval, which obviously has a lot of hormonal injections prior to the treatment of the woman. Fertilization would occur either with a needle and sperm through ICSI or regular IVF. The embryos are still cultured in what is still unlabeled culture media fluids. So it's quite strange that actually at the moment every food that we eat has to have the ingredients on it, and yet we can grow human embryos in a culture that we don't know the ingredients of. We know some of them, but the companies don't necessarily let you know. And then following this embryo culture, the embryos will be tested and then either vitrified or transferred for implantation. Another part of this is the idea of pre-implantation genetic screening, diagnosis, and testing. And I underline these because the term genetic screening has been somewhat overtaken because there is a new glossary of terms for infertility, and that is to incorporate the fact that screening is intended for a procedure used to determine whether the level of genetic risk for a couple is acceptable. And so the different screening that we would use for a pre-implantation genetic testing is quite distinct from pre-implantation genetic diagnosis, where you would be trying to diagnose a specific condition as opposed to screening for an array of conditions. So the new terminology for this is PGT, and this is further complicated by having individual names for the different procedures or what you would be screening for, and that is PGT-A for aneuploidy, M for monogenic disorders, and S for structural disorders. Further to this, again, just to remind you of the law that is involved in pre-implantation genetic screening and diagnosis, for every single condition that can be tested in PGD, we have a list of criteria, and they have a separate license has to be filed, and you can see on these whether they have been approved by the Human Fertilization and Embryology Authority or whether they are waiting. But each individual condition must undergo a separate application and be granted a separate license. So again, we can kind of mirror this very strategic and very effective process which we use for pre-implantation genetic diagnosis. And we've had 40 years of experience with dealing with genetic diagnosis, all the way from polymerase chain reaction, where we were just trying to look at one single piece of DNA, up to single-cell genomic diagnostics and whole genome sequencing. So we've really come a long way in terms of our understanding and our ability, rather, to screen and test and diagnose embryos. What's next on the cards is, regardless of whether you would consider these good enough, that you would actually have a method of screening or testing an embryo non-invasively. So what's a big problem that people probably have at the moment is the idea that you are doing something quite invasive to an embryo, no matter what. If you are taking a biopsy, it's an invasive procedure. So if we can move towards the point of non-invasive embryo screening, we'd really have something that would be actually much better than what we currently have in terms of testing an embryo. So the burden of genetic conditions is one major reminder of what it is that we are dealing with when it comes to germline genome editing. It's not a trivial thing. Somebody just spoke about the question of whether you would put a woman through something such as IVF. And I think it's important to remember that IVF as a procedure is not just isolating an embryo in a lab and growing it. There are a series of quite invasive procedures for the woman, less so for the man, that they would have to come together to do this. And it's a long process, but it's also quite a stressful one, both on the endocrine system and on the emotional system. So I think it trivializes IVF to assume that it would be done for something not considered to be a genetic condition. Nonetheless, with so many single gene disorders, and let's just talk about single gene disorders for now, there are progressions in the technology to say that we should be actually, certainly in societies where there are higher numbers of affected alleles, that we should be doing preconception carrier screening on these. And actually, previously, preconception carrier screening would look for just an allele, let's say cystic fibrosis, in a society where that allele was very prevalent. And now expanded carrier screening looks at an array of different genetic disorders that a couple may be susceptible to have when they come together. So I want to give you an example of, again, how this could be layered on top of a clinical pipeline. So in this example, which was actually presented at ASRM in 2014, 1,400 couples were tested for 190 common diseases. 30 of the patients were carriers of a disorder. Of these, 2% of the couples were carriers of a mutation in the same gene. So 40% of the couples who were both carrying the mutation underwent PGT. 50% of those undergoing PGT produced at least one affected embryo. That's a lot of affected embryos that actually were screened against due to the red flags that were signaled to them as a couple with their combined risk for certain genetic conditions. So maybe we can look at expanded carrier screening, certainly in certain societies, and say that it might be a proper approach. So this actually is the consensus guidelines for expanded carrier screening and what we should be testing. So the inclusion criteria for the disorders could be something that we could again mirror, and that would be that it would have a high carrier frequency among that ethnic group, that it would have a well-defined phenotype, have a detrimental effect on the quality of life, cause cognitive or physical impairment, require surgical or medical intervention, and early onset of disease in life. What hasn't been included in this consensus criteria is adult onset diseases or autosomal dominant conditions. Now, the problem of mosaicism has been mentioned, and mosaicism is what comes up time and time again at every single IVF and assisted reproductive technology conference. And that is because we have an increased ability to look at, oh, I changed it, sorry. We have an increased ability to look at embryos. And with this increased ability to look at embryos, we have more burden on us to do something about the information we are given. So a recent paper has come out asking about an evidence-based scoring system for prioritizing mosaic embryos and aneuploid embryos following pre-implantation genetic screening. And some of them say that an aneuploidy rate of above 20% should not be considered for transfer. But then there's a problem with that, in that even with whole genome sequencing, there's a cutoff threshold of below 20%, so that it would certainly some types cannot be picked up from screening alone. So I've realized they've uploaded the wrong slides now, because I can see the next one. So I might just ask you to pause and let me put them up, because the next point I wanted to make is quite important. Can I just swap screen? Yeah? Oh, maybe not, sorry. It's your fault, not mine. I could continue on, but I have slides that I purposely want you to. Let me just check that this is right. No, it's not. That's a shame. No. Okay. I will forge on. I'll try and remember what I had to say next. So these are a list of some of the procedures that are done in assisted reproductive technology, and they are no different to what would already be done. One of the slides that I wanted to go through was some of the individual both birth rates and the imprinting disorders, which we just spoke about. And that was that they have an increase in a lot of the prevalent imprinting disorders for babies born through IVF or assisted reproduction. And then the argument for this is actually to answer the question that it is not known whether it is the procedure of IVF itself or the patient group. So actually it is thought that infertility is probably the reason that you get this, this imbalance of imprinting in these patients, where they would have a higher prevalence of Beckwith-Wiedemann syndrome and an Prader-Willi syndrome and an Angelman syndrome. The next thing I wanted to ask about was consistency. So consistency is critical in the field, and what we are dealing with in human embryos are inconsistent beings. So IVF in itself is an imperfect technology. In fact, it's a collection of imperfect technologies, and it may never be a perfect technology because what it is working with is an even more imperfect and unpredictable medium, and that is human biology. So it may be that IVF combined with another imperfect technology, which is gene editing, may never reach the success rate that we would want it to achieve. And success rates are an important thing to remember, because when somebody asks me, what is my success of having a baby at the age of X? To me, it's almost like saying, what is my chance of getting better if I'm sick? You have to consider the individual in all of the cases, and it's the same with an embryo. You have to consider each embryo as a separate entity. And with the embryo as a separate entity, you have to include both parental genomes and how they're going to come together, the reproductive history of the patient, the age of the patient, because all of these are variables which are very rarely considered when it comes to success rates. The only true success you will have is a live birth, and yet earlier studies for success rates would be printed as those that got a chemical pregnancy or a clinical pregnancy, but they weren't actually presenting their live birth rates. And when IVF clinics started to get a rap on the knuckle for doing this, then they started to say, okay, we'll publish our live birth rates. But the patients that they started to take were under 35 and good prognosis patients. So again, you got a skewing of your success rates based on the individual clinic and the patients that they were accepting. So what we really need is consistency across all of the studies that we're doing. We know that human biology is inconsistent, but one thing that should be consistent is scientific rigor. So what I wanted to call for, actually, when people were saying, what do we do moving forward? Everyone talks about broad societal consensus, but actually among the scientific community, what we need is consistency and a dialogue that is an ongoing one. Somebody asked me, how do you keep up with all of the advancements? And I say it's actually very difficult. I started to save individual papers to tabs to read later, and I had 302 tabs at the end of the week that were very important reads. So I think that the scientific community should come together and start to dissect the technology. That is, that we don't say genome editing does or doesn't work. This base editing has off-target effects or not. That in fact, you look at the individual paper, the individual intended cut, the cell type, the endonuclease that was used, the method of delivery that was used. These are all individual variables that must be considered before we categorize a technology based on how good or bad it is. All of the different components must be dissected from a technology before we can say whether it's good or bad for clinical use. Thank you.
So for our last talk in the session, we're going to go back to more of an ethical discussion, or discussion about ethics, sorry, with Judith Daar, who's University of California, Irvine School of Law, also has a prominent role in the ethics committee of ASRM, I believe. Yeah, so okay.
Thank you. Well, good afternoon, everyone, and thanks for sticking with us to the very end. I'm so delighted to be here and have enjoyed this tremendously. And over the last 3 days, we've heard passing reference to the concept of informed consent in the context of research. So I'm delighted to have an opportunity, with my few minutes here today, to drill down on that and talk with you about the concept. And I think that it is self-evident to this audience that informed consent rests at the foundation of the legal, the ethical, and the regulatory framework that governs medical research in general and embryo and gamete research in particular. So it's essential that we do discuss it as a group. And I was reminded last night in Altacharo's wonderful session on research ethics about the need to consider the landscape on which we operate. And there was mention at that session about the simultaneous existence of innovations, research, and clinical application within our field. And I think it's fair to say that while those entities are distinct and yet they can be described very separately, that they really are unified by the concept of informed consent. And it has a rather stabilizing influence in a field that can be very unsteady at times. So that's a bit of a pitch as to why it might be essential to consider this. I also want to say before I proceed that my slides really address a research framework, but there's no reason why the concept couldn't overflow and be applied to a clinical setting, particularly with respect to IVF and that lovely talk we just had. So I begin then with this statement that's been mentioned here already that Dr. Collins issued in April of 2015 about the NIH position on, essentially on funding of gene editing technologies. And there's a longer statement, but I want to just draw your attention to the end of it, in which one of the reasons given for withdrawing federal funding or not making available federal funding was this idea that we don't know about some of the safety and ethical considerations, but also, reading 3 lines from the bottom, the way that affects the next generation without their consent, and so on. And I found that really fascinating, that the federal government is linking a decision about funding to the concept of consent, and not just consent about those who would participate today in the research, but rather those who would result from that research, the so-called future generation, and the impact that the research would have on them. And so linking funding to consent should remind us of the import of that concept. So what I want to do to approach this is to think really structurally about consent. There's a lot that we could say in the substantive arena, but I will defer. That is, we could talk about what needs to be revealed. We could talk about, do patients and research subjects actually comprehend those 25-page consents that they sign? And that's, of course, a very important matter. So I'm going to limit my remarks really to process-oriented concerns. When do we seek seek consent, from whom, from which body should consent be sought. So my talk is really very process-oriented rather than substantive, just as a disclosure. So I'll just begin with this notion of where we should seek consent. What are the sources for consent? And again, in the research setting. And I've identified 4, and there are probably others. Let me just talk about them very briefly. I think the main source for consent, particularly for embryos, are going to be from patients who underwent or are undergoing IVF. And the division there is to think about whether the embryos are provided by the intended parents through their own gametes or through donor gametes. And the role of donors in this arena is very essential. You might know that in the IVF setting for reproductive purposes, gamete donors, particularly egg donors, are occupying an increasing role in reproduction. You might know that the CDC demands that fertility clinics report their success rates each year, and one of the features, one of the metrics that they have to report are the number of births through donor eggs, donor oocytes. And currently, about 12% of all IVF babies are born in the United States with the use of donor oocytes. So it represents a very significant portion. And so there may be instances where embryos are cryopreserved or left over, and they're not needed, and there may be a desire to donate them down the road. And the question is whether the original donors, the oocyte donors, have to be consented, and at what point. Then we move on to gamete donors as a source of consent. And again, there's at least 2 ways that I'm identifying that donors come into the picture. They may be directed donors, that is, they may have been solicited by intended parents, either formally or sometimes very informally through social media and other methods, to provide their gametes. And the question is, should they be consented for the use of those gametes in a post hoc way, that is, after reproduction, but in a subsequent research setting. And then also increasingly, donors are providing their gametes to clinics or to banks without any immediate disposition that's been set out. And so the question is, should those donors be consented, and when? Also, clinics and banks are going to be the recipients of gametes and embryos, and they may be in a position to give consent for those materials to be used in research. There's a big debate within the IVF community about so-called abandoned embryos and gametes, particularly embryos at this point. We know from research that there is anywhere from 1 to perhaps up to 4 million embryos that are in frozen storage in the United States, over which many individuals are not exercising dispositional authority. So they might be considered abandoned, which is a legal concept per se, but that is just to say that there is no one claiming ownership or taking responsibility for their disposition. It's creating concerns in clinics, but it also may be of interest to the research community. ASRM takes the position that abandoned embryos should not be provided for research purposes, irrespective of any consent that the original providers may have expressed with respect to research. Also, increasingly with the banking community being so robust, there may be instances in which donors to these banks provide very generalized consent for disposition and then step back from having any authority over their gametes. And these donors, or the embryos that are formed from their gametes, may be so-called unselected. They may not be taken up by the market that really does patronize these banks. And the question is then, could the banks themselves provide consent for use in research? And finally, I have an idea I just want to chat very briefly about that really hasn't gained any traction clinically, but might be considered. And it's the notion of appointing guardian ad litems for gametes, and particularly for embryos. And this would be for purposes of consenting for their use in the research setting. So let me just speak about that for a moment. So in the law, there's a concept of guardian ad litem, which largely applies in the probate process and often in child welfare legal proceedings. And so I would take this model in the probate arena to describe how it might be used in research. And the way it works is that an individual, an adult, not always a lawyer, although sometimes, would be appointed to represent the best interest of the child. And in some instances, those children can be unborn. They can be imagined. And they may never be born, but they would have interests that could be protected in the legal arena. So just as I do with my class, I'm going to suggest a hypothetical. I promise not to call on you as I would my law students, but we would create a hypothetical to see how this might work. So imagine a situation in which a husband and wife have to undergo IVF because the husband is experiencing cancer diagnosis and is about to undergo chemotherapy. So in that situation, the husband might donate his sperm for protective purposes, protecting his fertility for the future. And let's further imagine that in that process that embryos are formed. Whether sperm is retained on its own isn't really as important to the fact that you'd have these embryos that are in frozen storage for a time when the husband recovers. And let's imagine further that the husband does not recover and that he dies. And he dies with a will. And his will provides for a trust to be established for his surviving spouse and any unborn children that might result from the embryos that remain in frozen storage. And this is a very common scenario. So in this situation, again, not to get too much in the weeds of the legal property aspects of it, but the will could provide that the wife would be able to use the estate of the husband for purposes during her life, but she would be required to preserve some of his assets so that they could be distributed and benefit the children that might be born after the wife has died. And this form of intergenerational wealth transfer is very common. So coming back to the guardian ad litem scenario, what would happen if the wife decided that she really didn't want her future children to have any of her assets, to have any of the money that was left by the husband? She could actually petition a court for a revision in his testamentary wishes, and it would be in that setting that the children or the unborn children's interests would be represented, and it would be a very common thing to transpire. Well, in that same fashion, I'm suggesting that we might consider a model in which a guardian ad litem is appointed to represent the interests of yet-to-be-born, unborn individuals in the research setting. And they would be charged, as guardians are typically charged, with representing the best interest of the children. Now, that might mean that research would not move forward, but at the same time, if the other considerations are taken into account, a guardian might very well approve their use in the research setting. And I confess in my slides that this is very problematic in the ART community, the notion of having unborn children in the form of embryos represented in a legal capacity. In fact, there have been a number of attempts on the part of interest groups to develop a guardian ad litem system. This has been noteworthy when there has been conflict among husband and wife in a divorce setting, in which there's been a suggestion that frozen embryos that are the subject of a dispute should be represented as to their interests. And this impacts on a number of features, personhood and other features. But it is still an idea that might be worth consideration. All right, just moving ahead. Let's talk about standards for approval. And again, we have to look to analogies and we have to look to existing areas of the law in order to know how to move forward, because we don't really have developed at this point standards that apply in the research setting. So what I have here are just the known standards that have been adopted at the federal level for research that is approved and typically is funded by the federal government. And again, not reading through this rather busy and wordy slide, what you see is that the considerations or the standards for permitting research look at a variety of features, including the risk that applies and the benefit that might be gained. So in other words, in order to consent for research using gametes or embryos, the question is, should we draw upon the literature and the regulatory scheme that already exists but applies to different types of entities? It applies to pregnant women, it applies to fetuses, and it applies to children, even very young neonates. And are those appropriate models to determine whether we should move forward with research involving embryos or gametes? It's hard to say, and those have yet to be developed, but I'm just suggesting that there are at least models for looking at and measuring the appropriateness of moving forward. All right, now I just want to move briefly to the question of when, and this is a debate I think that's been ongoing. When is it appropriate to seek consent from human subjects, either themselves or as proxies for their gametes and embryos when moving forward with research? And again, there aren't really solid answers to that question. At ASRM, we have a position statement that, as you can see, is somewhat outdated at this point. We will likely revisit it, but we take two positions within the document as to when consent should be obtained for research. So the first one, we say that it's important for patients to decide to donate their embryos only after they've decided not to continue storing them. So in other words, this is a post-treatment admonition that we suggest that it's only appropriate to solicit patients for embryos after they've concluded either treatment or their family, and that any prior request would be inappropriate. At the same time, you can see in the second statement, we address the donors, and we say that embryos that are created with third-party donors should be used to research only if specific consent for the use was provided by the donor in advance of the creation of the embryos. This is a very different point in the timeline in which we suggest that consent from the donor should be obtained. So we'd be asking for the donors to consent prior to the formation of the embryos, but yet we'd be asking for consent from the intended parents after their treatment is concluded. So as to timing, I'm just suggesting with this slide that we might want to rethink some of the principles that have been put in place in our documents and in other places with respect to the timing, and just trying to break up at least two time points for consideration. Is it appropriate to seek consent for research prior to retrieval? And again, I have listed here in bullet points the individuals and the instances in which it might be preferable to seek consent prior to retrieval. From patients that are using their own gametes, again, this goes a bit against the position that we take in the ASRM document. For known donors or directed donors, maybe it's appropriate to ask them prior to retrieval. And certainly in the research setting, that is very common and moves forward that the consent is, of course, prior to retrieval. On the other hand, what about consent after retrieval? And I want to just hint here at the bottom two bullet points under the post-retrieval scenario that currently, with the advent of cryopreservation, some of the consent forms that were developed and were used at the time that either the gametes were retrieved or the embryos were created have become somewhat outdated, that they don't necessarily address the kind of research scenarios that have moved forward in such a rapid fashion. So that's why I'm suggesting that it might be appropriate to rethink the pre-retrieval norm, because much of what was asked or what was suggested to the individuals wouldn't be clinically relevant today or wouldn't be relevant in the research setting. So we might want to give ourselves an opportunity to revisit consent based on emerging technologies and emerging research protocols. Again, just going a little bit deeper into the question of when, there's been, I think, a lot of advocacy for what I call contemporaneous consent for research. And some of this emerged out of the regulatory suggestions made during the Obama administration, that any research on embryos or gametes had to be secured by consent that was given at the time that the research was moving forward. And there was a lot of discussion within the the ART community about that matter. And I'm just suggesting here that perhaps revisiting this requirement for contemporaneous consent for research might be appropriate. And again, just posing 3 questions. Should contemporaneous consent always be required? And again, these are just scenarios in which perhaps it should always be required. Certainly direct donation to research, it makes sense to seek consent at the time the research is moving forward. Embryo donors after IVF completion, again, that's the traditional position, that even if individuals give consent prior to the formation of embryos, that it might be appropriate to check back, to double back and to ask, are you sure you wanted to provide consent? And I say this because today most of the clinics' consent forms have disposition provisions within the forms. And what they ask the patients is, if you don't use your embryos, and we list a group of contingencies: death, divorce, completion of your treatment cycle. And the query is, what do you want to happen to your embryos? And usually it's a check-the-box scenario in which you can indicate your preferences. And most forms have a box that provides for donate to research. And so if a couple or individual checks the donate to research box, the question is that once the treatment is completed and the embryos are no longer needed, do the researchers have to re-consent those patients for use of the embryos? And I'm just suggesting we might want to rethink that. Just moving along, what about the donor scenarios? And again, this can be somewhat problematic, because donors don't necessarily think that they will have any dispositional authority over their gametes after they provide them, either to the intended parents or to the banks. And so they don't anticipate the research question often. And so that raises the issue of, should researchers then have to trace the donors and seek their consent? Or are there other opportunities? I see that I'm going to just move along. And then finally, as to the question of the durability of consent that's given at any time, either before retrieval, upon the formation of embryos, or even after embryos are formed, I'm just suggesting that there might be ways to anticipate this question of seeking consent with either broad consent language, in which the forms would have general opportunities for research, what's a roll-down consent in which patients could check various things that they approve or don't approve, or even specific consent as to that contains project descriptions, source of fundings, and so on, which is actually very, very common today. All right, just to wrap up, in the context of the meeting today, there's been a lot of discussion about either embracing or rejecting harmonization in an international setting. And certainly consent is an area to be considered in this debate. And it seems to me, at least, that the global aspects of germline gene editing will impact the consent process. And I'm suggesting that it's best to plan for inclusion, that is, to think about the broadest and the most inclusive way to move forward so that it meets the needs of those operating in this space across the world. I'll end with that. Thank you so much.
I mean, maybe it's worth having all the panelists up here, actually, because I think there are going to be some questions that spread out. We're supposed to have a private discussion, so we'll start off with questions specifically for Judith and Helen, but then,
sure.
So we just to save a bit of time.
Do you want to bring the others up?
Yeah, if everyone.
I just want to, first, it was really extremely interesting. But I'm Hervé Schneves from France, and I'm the head of the French National Institute of Health Ethics Committee. I wanted to say that the discussion on informed consent is clearly an international discussion, but the way it goes takes place in various environments. And for example, in the French law, the bioethic laws, it's quite clear that what is happening to embryos are directly connected to the fact that it's at that stage the project of a couple. So, for example, if the couple break or if the husband dies, it's over. It's no more possible to use the embryos. There is a discussion now in the revision of our laws on the post-mortem implantation possibilities, but at that time it's forbidden. So the possibility of having guardians in the French context, for example, should not be possible because the destiny of the embryo is already decided. And it's quite the same for the consent. It's clear that for us it's anticipatory before the process, at the very beginning of the process of in vitro fertilization, the couples are informed of the various possibilities, but if the embryos are used, the leftover embryos are used for research, you need to go again for the consent because of the general principle that you can change your mind during the process at any time. So you need to have this contemporary re-consent, which is very often very difficult to obtain because when the couples decide to stop the process, either because they have a child or because it's a failure, very often they don't want to consent again, but it's the process. Thank you.
Well, I was actually going to ask, again, both in US context and UK context, can consent be withdrawn at any point, including after an experiment has started or after clinical use has started?
Yes, in general, the idea of rescission of one's consent is well accepted. But the problem I think you raise is this idea of presumed consent. And to what extent are we willing to embrace that? So the notion would be that if there's an initial consent that's provided at the time of retrieval, for example, can we presume its durability? And you're right, there's not a norm for that at this point, although we certainly embrace presumed consent in other contexts, so it might be something to consider.
I do know that in the UK that the original case, which was a woman who froze her embryos with her partner, and the reason they froze their embryos was that it was more than 10 years ago, so the ability to freeze an embryo, they would survive a lot more than freezing an oocyte. The recovery rates were too low because it was prior to vitrification, which was this flash freezing. And it meant that all patients now have to have quite considerable counselling beforehand to say what they would do with their embryos should one be deceased or the partnership break down.
Yeah. There's provision in UK law that says that you have to discard an embryo stored frozen after 10 years, whatever. Of course, it's now starting to cause problem for elective embryo freezing, where you're going to have lots of embryos that reach their storage limit before the woman is
especially in the case where younger women in their 20s are being told they should freeze their eggs while they're young, and then they still are young 10 years later.
Yeah. Question here.
Thank you. Maurizio Balistreri from Turin. If I understand well, you say in vitro fertilization is something complex, risky maybe. Do you think the same of sexual reproduction? I mean,
absolutely.
And which are the consequences of this? I mean, if also sexual reproduction is risky, dangerous, complex, what we have to do? This is the first question. And the second question, what do you mean for unborn children? Because I don't have children, but now I am a little bit worried. I could have unborn children.
I'm not going to acknowledge your second question, unless you've donated. As for the first question, actually, that was one of my punchlines, which unfortunately I didn't get to say. But if we were to try and legislate for IVF now, given our ability to scrutinise every different technology to such a higher level, the more we look, the more we find. And this is becoming a big problem for things like pre-implantation genetic testing and screening, because no longer are we just looking at chromosome 16, chromosome 21 for aneuploidy. We're looking at every chromosome and seeing that actually embryos are quite imperfect. But when you look at natural reproduction and the percentage of babies that are born with heritable disorders, then you'd probably say we wouldn't legislate for human reproduction either.
Yeah.
So
yeah, I mean, I can make a comment on that too. So, yeah, obviously there are human reproduction is very dangerous, and you could say, well, something that increases the spontaneous, the de novo mutation rate is having an older father. So where it essentially doubled. So it's, on average, it's about 40. New single point mutations each generation, but it doubles up to 80 if the father is older. So maybe you should ban old men from having children if you want to take that away. And of course, another issue about natural reproduction is if you really want to influence the genetics of your child, it's who you choose to have a child with is the most effective way of doing that. So. Natural reproduction has benefits and disadvantages. Guido.
Yeah, thank you. I have a question for the same speaker. I think you gave an informative and interesting overview of developments in assisted reproduction and the offering of ever more types of tests in different contexts, preconception, during PGD, the carrier mapping, PGT, PGS. I have a question with regard to the growing interest in introducing preconception carrier screening in assisted reproduction. This is a fascinating development, no doubt. At the same time, it's a little bit worrying. I'm now involved in the study, and we have a strong impression that
and this is in ways why, with genome editing, perhaps we need to learn from the lessons that assisted reproduction and the IVF community, the mistakes that they have made in allowing for techniques, for tests, and for certain additions to current clinical practice to just be added onto a treatment and being charged for it when there is no formal randomized control trial to prove efficacy to say that it will result in a bigger chance of you having a life.
We should worry, in a way.
It's a worry.
I have one small additional question. For this reason, I would argue that professional organizations like ASRM in the States or ASHRI in Europe should insist in recommendations that if clinics offer this type of screening, that the necessary condition should be adequate follow-up studies and evaluation studies. What are you doing, and in whose interest? Okay, you agree?
Probably something to do with. I absolutely agree that they'd have to be long-term studies on each individual. There should be, and starting with germline editing, that we would have to implement the exact same things. rather than letting the clinic where the patient walk out the door, they have to have maybe that they would sign up to say that they would be involved in ongoing trials.
Okay, thank you.
Any other comments from anyone?
Oh, I was just going to say that we have to balance this, and you mentioned this, against patient interest. As you know, at least in the US, it's a very laissez-faire oriented industry, and patients are very demanding of these technologies. And I'm not in any way suppressing the idea of regulation and follow-up study, just entering the metric of patient desire for this information.
Yeah, the problem is patients have no idea at all what they are demanding.
Either.
No idea.
None.
They don't realize the complexity of what they are demanding.
Yeah.
So informed consent, yes, tricky.
Tricky, yeah. Okay, we can start opening up questions for everyone, but let's go here.
I had a simple question. I was interested in the comment that IVF increases the rate of diseases like Angelman and Prader-Willi. Is it understood? Okay, small. I guess I was wondering how much and why, or how.
The overall rate is not an increased, is not a significant increase, but there is certainly higher percentages of at least 5 of the imprinting disorders among babies born to IVF or ICSI, and it's still debated whether that is as a result of the culture media, the process, or whether it is the parental origin of these births.
Yeah, it's not a huge increase. I think it's
I'll show you my slides
not at all.
afterwards.
It's only marginally significant,
yeah.
if at all. So they're still low.
Also, a well-known side effect, if you will, of IVF is multiple pregnancy, and although we're working to reduce those have certain morbidities associated with them. So that's always seen as an aspect of health.
I mean, to the other point, I guess I'm curious as to why it would be difficult to dissect that, because for those diseases, I think the genomic region that gets deleted
PGT, yeah.
PGTA.
Yeah, sorry. And so could something similar apply with genome editing, in that one of I think we understand that it's going to have to be done at a pretty early embryonic stage, and given that one of the situations in which we're thinking
embryo, that you would create the edit in every cell and it would be representative, and yet if what you're saying is that we have to wait to check whether it has the effect, then we have to wait till a later stage. I think this is going to be a twofold thing, where we will get better at probably doing a reversion of a later stage embryo, for one, and that if you had a very high percentage chance with expanded carrier screening of predicting, then maybe what you would put in is something like a conditional editing machinery to say whether it could detect the actual mutation. It was the guide RNAs could be guided to a specific locus dependent on the presence or absence. So if it wasn't there, that it wouldn't do anything, but if it was, it would be activated.
that would be a really interesting way of scientifically designing around a perceived ethical approach to risk, I will say. Thank you. Can I have a question for Judith? Or?
Yeah. No, go ahead.
Judith, thanks for your exploration of consent. I take it that most of what you were thinking about was consent to
they consented to being produced in the way that they have been. And I think we all recoil from that idea. And I've always urged my students in particular to think about that equality model. If we wouldn't regulate natural conception in the way that we're thinking about regulating assisted conception, then we need to push back. But that said, what you're suggesting is that it might be appropriate to think about the best interest of the future children in a more robust way than we currently think about the best interest of children that are conceived in a natural manner. And if you were to look at a guardian ad litem scenario in which the charge is to operate in the best interest of the children, it's quite possible that this would never move forward in any clinical way. It's quite possible that the decision would be very, very cautionary, as it is typically in those scenarios, and you wouldn't move forward. I'm not suggesting that normatively that's a positive or a negative, but if you truly looked at those values, they would apply in that fashion.
Thanks.
Kathy, are you going to ask about consent or something else? Because I want
something else.
Okay, can I ask one question about consent first, which is relevant to this week? Do you think the revelations that we heard in detail yesterday are going to affect the willingness of women to donate eggs or embryos for research purposes?
You never know how any individual takes in new information. And it can be surprising that things that we think might have very negative influences actually invite more participation. So perhaps there are members of the community who are reading about this across the world and are very excited by the prospect of at least the representations that were made, and in fact would invite more participation from women in research studies. I mean, I understand the instinct is to say that this is a debacle beyond description and therefore it's going to suppress research and volunteerism in the research community, but I'm never amazed and perhaps should be at the participation that it actually invites by some segments.
Thank you. Kathy.
I guess I'm asking a hypothetical, technical question. It seems that a lot of the time we have been discussing genome editing because of germline transmission, basically, of the mutation or whatever modification you've made. However, then there's been a lot of discussion about wanting to make sure that you didn't have mosaicism. But I just want to ask whether, in fact, having a mosaic person might actually be an advantage, because it then might mean that you cannot transmit the edited version of the gene through the germline. However, in some circumstances, for some disorders, maybe if you had a mosaic tissue, that may be sufficient to provide some kind of benefit. So I just would like to ask, if that's something that people might like to think about. It may be different for different disorders and different tissues, but is this a possibility?
Maria?
Yeah, so I actually brought that up very briefly yesterday. Even in the CCR5 case, if there was mosaicism, then there'd be a reservoir of cells that could not be infected, but then there'd be a reservoir of cells that could be infected, but if there was no infection, those cells maybe would provide resistance to West Nile virus. So even in that case, I mean, who knows what the germline would look like, but for that individual, maybe mosaicism has some value. I hadn't, until the CCR5 case, I hadn't really considered that, but the sorts of things like that Charlie was talking about, you don't have to have 100% correction, and so mosaicism may be sufficient, but I'm sure it raises all kinds of questions and concerns, since it's not so much a natural situation, although mosaic individuals do exist.
So sort of along those, related to the question about mosaicism, I mean, you don't have to do the genome editing at the one-cell stage either. You could do it later. And as Kathy mentioned, I mean, there's a very nice paper from Janet Rossant's lab which suggested that doing it at the two-cell stage can be really efficient. And in her paper, she showed that they could get homology-directed repair to insert a fluorescent reporter in 3 different genes at the same time, so in the same embryo, at very high frequencies. So that, you know, using clever tricks, but it was very impressive. You could also use perhaps at even later stages where you might want to use a viral vector. So we're not just limited to working with just the one-cell oocyte or embryo. And just to point out for those who may not have followed all the literature in this domain, that of course people have also developed techniques of doing in oviduct genome editing by essentially injecting the components into the oviduct. This is done in mice, into the oviduct, and then giving it electroporation and getting the components somehow to go into the embryo in oviduct. So you're not even talking about having to use IVF in this case. So that's a concern as well that needs to be thought about, because we can't have that fall outside any controls or regulations. So, question here.
So throughout the conference, a lot of people highlighted the potential concern of equitable access to germline editing. And it wasn't entirely clear to me if most people were thinking about somatic or germline, but one of the things that I was thinking about is that normal IVF per se is far from being equitable at the national and international level. So I think we should even consider that before we even start thinking about germline, I guess. But a lot of people mentioned the concern, but I didn't hear a lot of actual debate and opinions about what is going to be the impact. So I was wondering if you could comment on that. Do you think that coupling IVF with germline editing for the treatment of severe diseases will actually make it more or less likely that you know, governments and insurance companies will, you know, be willing to subsidize these interventions? Because I can see arguments both ways, but I would like to hear what do you think about it.
I think you have to make a distinction there between the United States and the UK, where actually it is both infertility treatment and IVF cycles are subsidised and are covered under the National Health System, and also PGD is covered for certain conditions, so that people will have free treatment, unlike in the States where everything would have to be covered by insurance. So there is, in some ways, you have to distinguish between different jurisdictions because the inequity is not the same.
Yeah, but I wasn't actually thinking specifically about the US or the UK. I mean, at the very global level, it's still, I mean, access to IVF is still very much a rich person's, you know, prerogative, or, you know, so I'm thinking at a very global scale. I'm not even thinking in an Anglosphere. But my question is, do you think that coupling that with genetic editing would make it more or less likely that governments and insurance companies would be willing to sponsor that?
I think it's an interesting point to make, but I would definitely take that if it was an older genetic editing technology. So we know that TALENs and ZFNs, a lot of money has been invested in those, and that's why a lot of companies are still proceeding to their clinical trials and to use them on patients, and that's because of the money that was invested. But those were a lot more expensive in their methods and in their means. CRISPR definitely could be argued to be the cheapest and most effective of any of those types of solutions. So I would say that it's not necessarily the practice that it would be the expensive part. Certainly IVF and the field of assisted reproduction is overly expensive.
I'm not arguing that, because, okay, to make it more clear, the point is, some countries, for example, the governments will say, or the insurance companies will say, we will not pay for IVF because you have alternatives. You can adopt, for example. But imagine a scenario where you naturally have a child, and that child has a genetic disease. The burden of healthcare of that child to society or to the state, the lifelong burden, will be very considerable. If the state has the means to avoid that by providing IVF and genetic editing of that embryo, you know, there is a financial argument that I think states would consider. So.
Well, you're assuming rationality in economic decision-making, which could be problematic. I mean, I think what you're suggesting is the risk-benefit analysis would suggest that governments and funders should go ahead and make the decision to fund at the input level rather than lifelong health concerns of the child. And again, I can only speak right now from a U.S. perspective to say that as rational as that is, we operate in the breach. That very few cycles of IVF are actually funded by the government, even though study after study shows that if there's government funding and reimbursement, then the rates of multiple pregnancy decline, the risk to the moms and the babies is reduced, and the costs are overall lower. And they're not, the differential, it does not reach the level that it would take to care for those individuals. That is, the price of IVF is much lower. And even though we've shown that in economic models over and over again, it just hasn't been taken up in a sort of a governmental way.
Okay, maybe this should be the last question. Yeah.
So we've talked about consent in the context of descendants, unborn descendants not being able to give consent. But I would argue that with germline, how I understand it, we don't only have to get consent from the direct descendants, because the changes they inevitably flow to the human gene pool. So who is the gatekeeper of the human gene pool? And do you think it is okay for people to create changes to the gene that inevitably will flow to the human gene pool? Thank you.
You know, again, I would suggest that we already have a scenario in which we might solicit consent down the road from lineal descendants. For example, when we have autosomal dominant mutations and we know that there's a certain risk of those presenting in generations down the road, we don't seek consent in any legal way from the children that might be born of those scenarios. So I'd be very cautionary about sort of creating exceptionalism in this arena when we already know that scenario is produced worldwide on a daily basis.
I mean, you also have what used to be called PGD, which of course is already current, and that is selecting embryos for a particular genotype. So that already occurs, and we've dealt with that okay.
We're also already doing, we're already, with certain IVF practices, creating heritable disparities or disadvantages to people. So ICSI, when you inject the sperm into the oocyte for fertilization, has now overtaken regular IVF to where there is before it used to have you used to have a clinical need to do ICSI, and that was male infertility, and now it's done just as a routine clinical practice because many of the embryos will go for screening and they don't want contamination of that embryo. And I would argue that for any male with male factor infertility that they were going to do ICSI for, then they should also have to, then they should possibly have to do sex selection for those embryos, because if that male who is infertile has a Y chromosomal deletion, well then you are doing ICSI and you're basically conferring infertility to the next generation. So any son born to that ICSI procedure would have the exact same Y chromosomal deletion as the father. So we're already doing transgenerational changes which are probably unwanted.
And it's of course the partner of that next generation would have to undergo the IVF procedure.
It's a very good business model.
Okay, I think we should call it an end. But please, can you thank the panel for a very good talk.